Good morning. I would like to welcome everyone to today's MD Anderson Cancer Center Enjoy Science seminar. Today is the start of the summer program with weekly presentation on Fridays. This is a series of lectures given by MD Anderson faculty members and organized by the Office of the Chief Scientific Officer, Dr. Giulio Draetta. I'm Alessandra Ferraioli and have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of the Enjoy Science initiative is to strengthen the connection between the scientific and medical community at MD Anderson Cancer Center and our colleagues, our patients, and our friends around the world during this time of limited in-person at the bench and in the clinic activities. Today's seminar is being recorded and will be made available on the Enjoy Science website later next week. The speaker of today is Dr. Kati Rezvani. Dr. Rezvani is a professor in the Department of Stem Cell Transplantation. She also holds other titles as the medical director of the GMP Cellular Therapy Facility, director of Translational Research, and section chief Cellular Therapy, all in the Department of Stem Cell Transplantation Division of Cancer Medicine at the University of Texas MD Anderson Cancer Center. Dr. Rezvani is a world-known authority in the field of cellular therapy. She has developed innovative approaches in this field and will be discussing for us the topic of CAR NK cells, a drive to the future of cell therapy. So without any further delay, we're going to hear Dr. Rezvani presentation.
Well, good morning, everyone. Thank you very much, Alessandra, for this very kind and generous introduction. And I'd like to thank you and George for inviting me to give a seminar this Friday. So the focus of my presentation will be on NK cell therapy. And just before I start, a number of disclosures. The one that's perhaps most relevant to this talk is that MD Anderson entered a license agreement and research agreement with Takeda to develop cord blood CAR NK cells for the treatment of B-cell malignancies and other cancers. Having said that, the data that I'm going to present to you was not sponsored with them, and it kind of predates really this agreement with Takeda. So what are chimeric antigen receptors? I'm sure most of the listeners know what CARs are, but just to remind people, a chimeric antigen receptor, as the name suggests, is a chimera. It brings together two elements from antibodies and T cells. So basically, it brings together the antigen recognition site of a monoclonal antibody together with the signaling and killing machinery of T cells, so that when a chimeric antigen receptor is inserted into an immune cell, such as a T cell or a natural killer cell, it has the capability to recognize an antigen expressed on the surface of a tumor, just like an antibody would, but we give the cell a signal to kill and really sort of harnessing the machinery of the killing machinery of a T cell or an NK cell. Let me just change that to laser pointer so that people see. Okay, and I'm sure there's a lot of kind of knowledge and understanding of CARs and really a lot of excitement in what CAR T cell therapy has done for the field of cell therapy. And just for those people who are not in the clinic or may not have been exposed to the overall patient flow of CAR T cell therapy, so you can see here what happens is a patient usually comes, they have their blood collected either through a blood draw or through an apheresis. And this product is then sent to a lab where the T cells are separated, usually using beads that bind CD3 on the surface of T cells. These T cells are then expanded in a specialized lab. They're activated, they're transduced with a viral vector, and then at the end of 2 to 3 weeks of expansion, a product is available. which can then be given to a patient after the patient receives lymphodepleting chemotherapy. So the product usually, in the majority of cases at least, is a patient-specific and autologous product. And really, the success of CAR T-cell therapy was first reported in the setting of B-cell malignancies. So this data summarizes 3 of the largest trials of CAR T-cell therapy targeting CD19 in patients with acute B-cell leukemia. And as you can see, these 3 studies were led by different groups. So this study was led really by the group at the University of Pennsylvania, although it was a multi-center study. The CAR product was a lentivirus-based CAR, and the co-stimulatory domain was 4-1BB based. This one was led by the Fred Hutchinson Research Cancer Center. Again, here they used the CAR-19, which was lentivirus-based with a co-stimulatory molecule that was 4-1BB, and the study that was led by Memorial Sloan Kettering used the retrovirus, and the co-stimulatory molecule was CD28. But as you can see here, the responses were pretty even amongst the different studies, and in the region of 80 to 90%. And this was pretty incredible because many of these patients were multiply relapsed, and many had even failed an allogeneic stem cell transplant. So these patients had a very poor prognosis. And really these data led to a paradigm shift in the field of cell therapy. Many of these responses, as you can see here, were persistent, and even up to a year after treatment, around half of the patients were still in remission. And CAR T-cell therapy for the treatment of young adults and children with acute lymphoblastic leukemia is now FDA approved, but it comes with a pretty hefty price tag of $500,000, and I'm going $475,000. So I'm going to come back to that shortly. Another disease that also expresses CD19 and that was shown to be also susceptible to CAR-19 T-cell therapy was non-Hodgkin lymphoma, B-cell non-Hodgkin lymphoma. Again, these are 3 of the largest studies. The ZUMA-1 study, which tested Yescarta or Axi-cel, was led by Dr. Neelapu here at MD Anderson. But again, you can see different viral vectors, different co-stimulatory molecules. The response rates were not quite as high as what was observed with acute lymphoblastic leukemia, but still it was very close, and you can see again the 1-year survival of these patients was around 35 to sort of 40%. And also CAR T-cell therapy for the treatment of non-Hodgkin lymphoma using this product, Yescarta, which is a Kite product, has been approved by the FDA, and here the price tag is around $373,000. So why is it that these products are so expensive? Well, as you saw from the workflow that I showed, they are patient-specific. So making a product for a patient specifically that then cannot be used to treat other individuals certainly then adds to the cost of manufacturing and certainly the viral vector that's being used. In addition, the studies of CAR T-cell therapy really kind of unraveled a toxicity profile that was not previously described with other types of treatment. And these were the cytokine release syndrome and neurologic toxicities. So what happens is when these CAR T cells are infused into patients, they are obviously very activated T cells. They see the antigen, they're highly potent. And through this, their in vivo activation, the CAR T cells themselves produce a lot of inflammatory proteins such as TNF-alpha, interferon gamma, interleukin-6, but also the CAR T cells crosstalk with other immune cells in that microenvironment, and especially myeloid cells such as macrophages, etc., activating them and leading them to produce other inflammatory proteins. And at least the syndrome of cytokine release syndrome is believed to be mediated by high levels of inflammatory cytokines such as interleukin-6 or TNF-alpha. And what happens is these patients become sick, they drop their blood pressure, their heart rate goes up, they drop their oxygenation, and may require treatment in the intensive care unit. And in fact, up to 30 to 50% of patients that receive CAR T cells end up requiring at least a day or longer in the ICU for the management of cytokine release syndrome. Deaths have been reported, although now they're rare, especially since the use of an anti-IL-6 antibody called tocilizumab to manage those symptoms. The neurotoxicity associated with CAR T cells is a unique syndrome. The pathology of it is not very well understood. And here, patients develop confusion, tremor, and in very severe cases, again, encephalopathy and seizures. This is also usually self-limiting and may or may not happen in conjunction with cytokine release syndrome. And often these patients, in addition to getting tocilizumab, they receive also corticosteroids. They can also dampen down the inflammation. Okay, so why natural killer cells? I've already shown to you CAR T cells have really resulted in a paradigm shift in the field of cell therapy. Excuse me, the results are pretty impressive. And sure, they are expensive and they have toxicities, but they're manageable. So we started thinking about natural killer cells as an alternative, partly because they have certain characteristics that may make them also advantageous for cell engineering. So NK cells are part of our innate immune system. Their phenotype is characterized by expression of CD56 on their surface. They don't express CD3. Unlike T cells that differentiate in the thymus, NK cells differentiate in the bone marrow, and they primarily circulate in the peripheral blood. They do not require prior antigen priming. And what is really unique about NK cells is that they don't cause graft versus host disease. Allogeneic T cells, even if HLA matched with the recipient, carry the risk of graft versus host disease because through their T cell receptor, they can recognize even minor histocompatibility antigen mismatches between the donor and recipient. And that's why in the setting of bone marrow transplant, allogeneic stem cell transplant, we always, in addition to matching the donor and the recipient at HLA molecules, we also have to use immunosuppressive drugs to prevent that graft versus host disease to happen, which is a devastating disease that if severe can lead to mortality. And, you know, that's one of the reasons also with CAR T cells, at least the FDA-approved CAR T cells that are in the clinic, and the majority of CAR T cells that are being tested in various studies are autologous, they're recipient-derived. NK cells, because they don't cause graft versus host disease, have the advantage that can be used from an allogeneic source. So that means from a donor that is unrelated to the recipient. I mentioned NK cells don't require prior antigen priming, and that's because NK cells have a capability to recognize abnormal cells through a series of germline encoded receptors that's on their surface. And so as part of the innate immune systems, NK cells are a first line of defense against cancerous cells or virally infected cells. So what I mean by these receptors is kind of presented here schematically. So this is an NK cell. I don't want you really to worry too much about the name of the receptors. It's not important. What is important to realize is that NK cells have these germline encoded receptors. So these are receptors that we're born with. And these receptors are either activating or inhibitory. The ligands for the inhibitory receptors are HLA class 1 or HLA class 1-like molecules. The ligands for many activating receptors are not known, but the ones that are described are stress molecules, which are basically DNA damage molecules that are expressed on the surface of abnormal cells after they become cancerous or they become virally infected. As the name suggests, the inhibitory receptors, when they bind to HLA class 1 molecules, give a negative signal to the NK cell not to kill. The activating receptors, when they bind to these activating ligands, these stress molecules, give a positive signal to the NK cell to kill. And ultimately, the decision of an NK cell to kill or not to kill is based on the balance between these activating and inhibitory signals. So as shown here schematically, when an NK cell comes into contact with a normal cell, and as I'm sure you know, all normal cells other than red blood cells express HLA class 1 on their surface. So as the NK cell comes into contact with this normal cell, the inhibitory receptor on the surface of the NK cell binds HLA class 1, and this gives a negative signal to the NK cell not to kill. In the setting of cancer, where often the cancer cells downregulate or lose HLA class 1 expression, this binding of the inhibitory receptor to the HLA class 1 molecule doesn't happen. Therefore, there is no negative signal. And now the activating receptor on the surface of the NK cell can bind the activating ligand, the stress molecule that I mentioned. The NK cell gets a signal to kill and then starts releasing cytotoxic molecules, etc. So based on what I've said, you can see why NK cells could potentially be perceived as a holy grail of cell therapy for immunotherapists, because NK cells have the capability to kill cancer cells, but they don't cause graft-versus-host disease. So just to summarize, at least our perception was that if we use NK cells for CAR therapy, well, because they don't cause graft-versus-host disease, now we can use an allogeneic product that's off the shelf that could potentially lower the cost of manufacturing, lower the cost of the therapy for patients, and I will show you shortly that we've demonstrated that from one donor. In our case, we've been focusing on using cord blood as a source of cells for cell therapy. For CAR-NK manufacturing, we can make hundreds of doses of cells. With CAR T cells, because of the risk of graft-versus-host disease, the product is usually autologous. So it's one product per patient. Having said that, there are a number of companies as well as a number of academic institutions now that are looking at strategies to use allogeneic T cells for CAR engineering. Here, however, you have to add one extra step of cell engineering, and that is to knock out or to delete the T cell receptor so that you remove the risk of graft-versus-host disease. So we discussed the GVHD issues, and also, you know, NK cells have those activating receptors on their surface that I mentioned. So they have this innate capability to recognize tumor cells. So one could argue that potentially the risk of relapse with CAR-NK cells could be less than CAR T cells, and that's because whereas with CAR T cells, the main mechanism of tumor recognition is through the chimeric antigen receptor on the surface of T cells. With CAR-NK cells, in addition to the CAR, those germline-encoded receptors of NK cells can also recognize the tumor, so there could be synergy in tumor recognition. I've mentioned the toxicity profile of CAR T cells. NK cells have a different cytokine profile, so again, we hypothesized that potentially the toxicity profile with CAR-NK cells could be different to that of CAR T. But of course, NK cells have shortcomings. Otherwise, we would have treated, and cured all patients with cancer with NK cells. We've known about NK cells since the mid-70s, and some of the shortcomings are listed here. So unlike T cells that can persist really for the life span of a person, NK cells have a short lifespan. So mature NK cells only only have a lifespan of around 10 to 15 days. Now, there's some data, at least in animal models, that memory NK cells also exist that are long-lived. But I'm talking about conventional NK cells that only persist for about sort of 10 to 15 days. I mentioned that NK cells, through their germline encoded receptors, can recognize tumors, but not all tumors are susceptible to NK-mediated killing. Not all tumors express on their surface the ligands that the activating receptors of NK cells can recognize. So in certain settings, antigen specificity could be desired. For instance, NK cells are not very good at recognizing and killing cells of B lymphoid lineage, such as acute lymphoblastic leukemia, CLL, non-Hodgkin lymphoma. On the other hand, NK cells are very good at recognizing myeloma cells, glioblastoma cells, etc., which I will show you later. And also, NK cells are numerically fewer than T cells. So somewhere around only 5 to 15% of our total lymphocyte population comprised of NK cells. And of course, that's not sufficiently this is not enough number of cells for cell therapy. So we need to come up with ways of, first of all, finding a donor, and second of all, finding a way to expand these cells. And so different groups are using different sources of NK cells for immunotherapy. Autologous NK cells are not effective for immunotherapy. There's a lot of data showing that NK cells of patients with cancer are dysfunctional, which I don't have time to go through today. Some groups are looking at NK cell lines or differentiating NK cells from hematopoietic stem cells or induced pluripotent stem cells. Our group is interested in using cord blood as a source of NK cells for immunotherapy. That's for a number of reasons. We're very fortunate at MD Anderson to have our own cord blood bank, which was established by my colleague and friend, Dr. E.J. Spall, when she joined the institution back in 2003. So our cord bank has now collected over 80,000 units of umbilical cord blood that were donated altruistically by women who are giving birth in our collaborating OBGYN hospitals. And also, we've treated now over 50 patients with very high doses, sort of up to 10 to the 8 cord blood NK cells per kg. And we've shown no toxicity, so this is a very safe approach. And these cord blood NK cells were mismatched to the recipient. So we already had this platform of ex vivo expanded cord blood NK cells, and we decided to build on that and see whether we can engineer them for cell therapy to further kind of enhance their activity. The other reason why we believe that cord blood may have advantages over peripheral blood is when that we went and did a transcriptomic profiling of NK cells that were selected from cord blood versus peripheral blood NK cells, and then we observed that these cord blood NK cells have higher expression of genes related to cell cycle, cell division, and DNA replication, suggesting that maybe these kind of naive and immature NK cells from cord blood are more capable of proliferating and persisting in vivo after adoptive injection. So then we collaborated with Pietro Dotti at Baylor College of Medicine when he was there, and now he's moved to University of North Carolina to test a retroviral vector that was targeting CD19, so it had the single chain variable fragment against CD19. Signaling was through CD28 and CD3 zeta. This vector also had the gene for a human interleukin-15, so that cells transduced with this vector would continuously secrete low levels of IL-15, and IL-15 is a cytokine that is important for NK cell proliferation and persistence. And we argued that this could be beneficial because it could help with the in vivo persistence of the NK cells. And in addition, this factor encoded for a safety switch, so a gene for inducible caspase 9 that could be activated with the addition of a small molecule dimerizer called rimicid that is provided by Bellicom, and then would result in activation of the caspase. Senior scientists in our group developed the protocols for the transduction of NK cells with this vector, and we called that an armored CAR because it had the cytokine gene. And she then demonstrated that if you infuse now these CAR transduced NK cells into immunodeficient mice that have been engrafted with a human lymphoma cell line, a Burkitt's lymphoma cell line called Raji, now these CAR NK cells can be detected in the blood of the animals for 70 days or longer after infusion. As I mentioned, non-transduced NK cells, once you infuse them after about a week or two, then they're no longer detected. And this also translated into better in vivo anti-tumor control. So now you can see this is a very aggressive model. The NSG mice that get the Raji tumor here by bioluminescence imaging. About day 14, they have the tumor. By day 28, they're all dead. The animals that receive non-transduced ex vivo expanded NK cells, they succumb to disease by day 28. Again, there's no control of the disease. NK cells that are transduced with CAR19 but without R15 only control the disease transiently, but then the tumor comes back, and that's because the NK cells don't persist. But animals that receive the CAR19 R15 transduced NK cells can control the disease nicely. So then with our GMP facility, we developed the protocol for ex vivo expansion and transduction of the NK cells with the vector that I just showed you. So we made the GMP grade retrovirus, and here you can see we get a frozen cold unit from our bank. Our GMP facility thaws the cells, depletes the T cells, monocytes, and B cells. Here the negatively selected NK cells are then expanded and basically activated with a K562 based beta cells that are irradiated, and these are engineered to express membrane-bound R21, 41BB ligand, and CD48, which are two co-stimulatory molecules for NK cells and IL-2. On day 6, we transduce the NK cells with the retroviral supernatant. We do one more round of expansion with the beta cells and IL-2, and day 15, we have our cells. And these are data generated by our GMP facility. You can see that we're using 5 different cord units. And you can see from one cord, we can get somewhere around 20 to 50 million NK cells. At the end of 2 weeks of culture, we get around a 2,000-fold expansion. The transduction efficiency of the CAR is around 65%. And from one cord, we get a median of 44 billion cells. This then means that if we were to infuse a dose of 1 million CAR NK cell per kg to a patient from one cord unit, we can get around a median of 440 doses of CAR NK cells, really increasing accessibility and reducing potentially the cost of therapy. So then with these data, we went to the FDA and we got approval for this first-in-human phase 1/2 study of allogeneic cord-derived CAR NK cells. This is the schema of the trial. So first, we picked the cords to be partially matched with the recipients. That was an FDA requirement. But as we demonstrated the safety of the approach, now we're picking actually the cords based on no HLA matching. Where possible, we pick a cord that's clear ligand mismatched with the recipient. So the patients come in, they receive lymphodepleting chemotherapy with cyclophosphamide and fludarabine, the same doses for the autologous CAR T cells. In the meantime, our GMP facility expands the CAR NK cells using the schema that I showed you. Because this was a first-in-human study, first we did a dose escalation, where patients received 1 dose of CAR NK cells at the 3 dose levels here, starting at a very low dose level of 100,000 CAR NK per kg. Dose level 2 was 1 million CAR NK per kg, and dose level 3 was 10 million CAR NK per kg. So these data were recently published in the New England Journal of Medicine, so I'll go through the clinical data quickly. We published the the data from the phase 1 portion of the study, where we've treated 11 patients, 5 patients with chronic lymphocytic leukemia and 6 patients with non-Hodgkin lymphoma. Median age of the patients were 60 up to the age of 70. Patients all had very bad risk disease, very bad risk cytogenetics, and they had failed multiple lines of treatment up to 11 lines of therapy. The CLL patients had all failed ibrutinib. 2 of them had Richter's transformation. Some had failed also venetoclax, and the lymphoma patients had either all failed a stem cell transplant, or their disease was so aggressive that they had never quite reached a stem cell transplant. You can see here that actually none of our patients developed any cytokine release syndrome or neurotoxicity. And even the 2 patients in the highest dose level, where they received a cord NK cells that were completely HLA mismatched, there was no cases of graft versus host disease. Of the 11 patients that we treated, 8 of them had responses, and 7 of these were complete remissions. 1 patient had a complete remission of his Richter's transformation, and Richter's is a high-grade transformation of chronic lymphocytic leukemia. The patient continued to have the low-grade CLL in his bone marrow, so we call this patient actually a partial remission. And this is the patient here that you can see at a dose of 1 million CAR NK per kg. You can see this is a PET scan where the highly active tumor cells take up the disease, they take up the tracer, and you can see this patient had evidence of activity in his neck, in the axilla, and in the abdomen. This is the brain and the bladder that normally take up the dye. And at day 30 after CAR NK infusion, you can see all the lymph nodes have gone. This is just the patient's kidneys. And what's also important to note was that we saw responses at all 3 dose levels. This is a site of analysis performed by Rafet Bashar in the lab, who's a very talented postdoc, soon to be a faculty in our department. And so CyTOF allows us to look at 40 markers on cells simultaneously, so it's kind of mass cytometry. And you can see here, Rafet has categorized the different cells in different colors. You see this patient at day 0. So this patient had already had the lymphodepleting chemotherapy. The day 0 is the day he was scheduled to receive the CAR NK cells. He had a large population of B cells, very few NK cells here in red, and the T cells are shown, the CD4 T cells are in pink and CD8 T cells in purple. 6 days after infusion of the CAR NK cells, all the B cells are gone. And now you see a nice expansion of NK cells, and these are the CAR NK cells that we've infused. The T cells are still there, so you can see that the effect of CAR NK cells is very specific to the target, and it's only depleting really the CD19 expressing B cells. This is a busy slide that shows you the persistence of the cells over time, and I'll take you through it. So by qPCR, we quantified, and Lou in the lab quantified the expansion and the persistence of the CAR NK cells over time here. So the patients are categorized, shown here in different colors, based on the dose of the NK cells that were infused. In the peak of the expansion, as you can see, was in the first 2 weeks after CAR NK cell infusion, and the higher the dose that was infused, the higher the peak of expansion, not surprisingly. Beyond day 14, however, we saw that the CAR NK cells was present in the peripheral blood of the patients at the same level. And this suggested to us that actually the CAR NK cells expand in vivo, and even with these low dose levels, basically they expand and they find a steady level. But what was most surprising and interesting to us was that these allogeneic NK cells were still detectable 6 months and even up to a year. And now beyond the year, we've shown that we can still find these CAR NK cells at low level, which suggests that there is a degree of tolerance and chimerism that's being established, because remember, these CAR NK cells are allogeneic, so they should be rejected by the recipient T cells when they recover from the effect of the lymphodepleting chemotherapy, but these CAR NK cells are persisting. And certainly, you know, the IL-15 is helping with their in vivo expansion, as you can see. So just to summarize this portion of my presentation, we've demonstrated that NK cells can be engineered to express a CAR to redirect their specificity and to express a cytokine to enhance their in vivo proliferation and persistence. And in our first in-human clinical trial, we've demonstrated really very impressive, at least early clinical responses with no evidence of CRS, neurotoxicity, or GvHD. So 2 patients progressed and relapsed, and both of these patients had CD19 positive disease at the time of relapse. So we wanted to then ask, you know, what drives relapse after CAR NK cell infusion? So to answer this question, we went back to our in vivo mouse model, and Lily, who was a postdoc in my lab at the time, collaborated with Dr. Ken Chen in the Department of Bioinformatics, who's a great collaborator of ours, and 2 of his graduate student here and his postdoc, Jin Du and Chi Miao, to really look at the single cell level of NK cell evolution over time. So using our in vivo mouse model, where Lily injected the Raji cells and NK cells, and this is not a therapeutic model. So this, what we show is that the CAR NK cells prolong the survival of the mice, but eventually the mice all will develop disease and will die. Lily at specific time points sacrificed the animals, and then she harvested their organs and collected NK cells and Raji cells and performed single-cell RNA sequencing and CyTOF to really understand the in vivo evolution of the cells. And she made a very interesting observation, and that's kind of summarized here, that over time, CAR NK cells start upregulating molecules related to maturation and also to checkpoints, such as Siglec-7, TIGIT, CD57, and many of the activation markers that are important in NK cell cytotoxicity and function are reduced over time. So the question then was that, okay, can we then enhance the function of CAR NK cells by targeting checkpoints? So the idea that exhaustion and checkpoint activation it can contribute to failure of CAR therapy is not novel, and that's already been shown with CAR T cells. But of course, we wanted to see if that's the case with CAR NK cells. So one checkpoint that we were very interested in, and this was a study that was led by May Daher in my group, and May is now a faculty in our department, was to study this checkpoint called cytokine-inducible SH2 containing protein CIS. The reason we were particularly interested in this checkpoint is that this becomes activated, this protein that's a member of the FOX family becomes activated when cytokines bind to their receptors and activate the JAK-STAT pathway. There is activation of this SOX family And this acts as a negative feedback loop, really to prevent the sort of uncontrolled activation of JAK-STAT pathway in response to cytokine stimulation. And remember, our vector had interleukin-15 in it. So then we wondered if we delete the gene encoding for cis, and this gene is called CISH, can we enhance the activity of CAR NK cells? And this idea was not completely novel, because actually Nick Huntington's group in Australia had already shown that in mouse NK cells, deletion of CISH increases the hypersensitivity, makes the NK cells hypersensitive to the effect of IL-15. So what May did was, first she looked at the expression levels of CISH in CAR and 19IL-15 NK cells, and she demonstrated that over time, CISH levels increase in these CAR NK cells. And then in collaboration with Rafet and Elif Gökdemir and Nedima Upreti in the lab, they developed the protocols for combined CAR transduction and CRISPR gene editing using Cas9 and gRNA ribonucleoprotein. And here you can see by a PCR very nice knockout of the CISH gene with the two bands here. And certainly you can see here how the cis protein, by Western blot, we can see that the cis proteins practically being its expressions being downregulated and ablated. And then May went and tested the activity of the CISH knockout CAR19IL-15 NK cells in a non-curative Raji lymphoma mouse model compared to CAR19IL-15 NK. And now you can see that these CISH knockout CAR NK cells are capable of curing at least a proportion of the animals with very nice eradication of the tumor. So in order to then understand the mechanism, what May did was she did RNA sequencing, and she demonstrated, as expected, that the JAK-STAT pathway is upregulated, or the genes related to that. But also, she made another interesting observation in that CISH knockout in CAR NK cells results in upregulation of genes related to glycolysis, to the mTOR pathway, and also to c-Myc, suggesting that maybe part of the reason that these CAR NK cells are functioning better could be that they're becoming more metabolically fit. And indeed, when she then performed a seahorse assay to measure the glycolytic potential of the cells and the acidification rate, she demonstrated that here in red, the CISH knockout CAR19IL-15 NK cells have a better improved glycolysis when you compare them to the CAR19 NK cells that only have the Cas9 protein without knockout of CISH. And here you can see that that also in terms with the p-values. And what also she demonstrated was that then when you put these CISH knockout CAR19 NK cells in culture with the Raji cells compared to CAR NK cells that are wild type, there is higher activation of phospho-AKT, the mTOR pathway, phospho S6, and also c-Myc. And so based on these data, we think this is the model by which CISH knockout can enhance the activity of CAR NK cells, so that when we silence CISH and remove the inhibitory effect of the cis protein, now the binding of IL-15 with IL-15 receptor results in activation of AKT, activation of the mTOR pathway, c-Myc, activation of glycolysis, and basically the metabolically more fit NK cells. Okay, so I've shown you how CAR NK cells can be applied for the treatment of hematologic malignancies, but can we extend CAR NK cell therapy beyond lymphoid malignancy? And can we target glioblastoma? So around 4 or 5 years ago, we started a collaboration with Amy Heimberger and more recently also with Fred Lang to really try and understand the role of NK cells in the immunosurveillance of glioblastoma. So with glioblastoma and solid tumors, the situation is more complex because you have to also take into account a very hostile microenvironment, and also the fact that your NK cells have to traffic to this kind of solid mass of cancer cells. So Hila Shaim, at the time, she was a postdoc in my lab, and now she's doing her residency at UTMB, started this project in collaboration with Amy and her group to really look at whether NK cells are susceptible to, whether glioblastoma cells are susceptible to NK-mediated killing. And we were especially interested in the susceptibility of glioma stem-like cells, or GSCs, to NK cell-mediated killings, because GSCs are really the drivers of NK cell of GBM resistance and GBM relapse after therapy. They have tumorigenic potential, but also very importantly, they're chemotherapy and radiotherapy resistant, and this is GSCs that really cause and result in the relapse of patients. So what Hila did was she looked at the expression of different activating and inhibitory ligands on the surface of GSCs compared to normal astrocytes, which are the kind of normal component, normal counterparts. And what she demonstrated was, very interestingly, that GSCs actually express a lot of ligands that NK cell activating receptors can recognize, such as MICA, MICB, ULBP, which are recognized by NKG2D, CD122 and CD155 that are recognized by DNAM, HLA-E that's recognized by NKG2C. And also, and she confirmed that by doing a chromium release assay, and she demonstrated that allogeneic NK cells can kill GSC targets very well, the same as they would kill K562 targets. And K562 cells are always they're leukemia cell line that don't express HLA class 1, so they're always used as the positive control for NK-mediated killing, because they're exquisitely sensitive to NK-mediated killing. So you can see GSCs are killed nearly as well as K562 targets. Astrocytes, on the other hand, are not killed by NK cells. And then we collaborated with Amy, Daniel Zamler, and Ken Chen to study the tumor-infiltrating NK cells in glioblastoma, and then to compare them to healthy control NK cells and peripheral blood NK cells. So Daniel had already performed single-cell RNA sequencing of the immune cells within the primary glioblastoma tumor from patients, and he was kind enough to really share the data with us so that we could specifically look at natural killer cells. And how you can here in these t-SNE plots, you can see that the tumor infiltrating NK cells really separate very nicely from healthy control NK cells and from the peripheral blood NK cells of glioblastoma patients. So here again, you can see these are peripheral blood NK cells from healthy controls, peripheral blood NK cells from glioblastoma patients, and tumor infiltrating NK cells. And you can see that these tumor infiltrating NK cells are very different. They don't express many of the activation markers, or down regulation of many of the activation markers that are important for NK cell function. They have upregulation of many of the inhibitory markers, and also they have upregulation of genes related to the TGF-beta pathway. So this really gave us a clue to look at the influence of TGF-beta on NK cell function in glioblastoma. So what what Hila did was she took these tumor-infiltrating NK cells from primary brain tissue, and she asked whether these NK cells are capable of killing GSCs. And as you can see here, compared to peripheral blood NK cells from glioblastoma patients and healthy control NK cells that can kill GSCs, that can kill this is K562 targets. The tumor-infiltrating NK cells are dysfunctional, and they're not at all capable of killing K562 targets. And taking the clue of the role of TGF-beta should look at the activation of the SMAD pathway, which is the canonical signaling pathway downstream of TGF-beta. And what Hila demonstrated is that tumor-infiltrating NK cells have activation of the TGF-beta pathway, the SMAD pathway. So the question was then, are these GSCs inducing NK cell dysfunction through TGF-beta? So then what Hila did was she took GSC cell lines, cultured them with NK cells in the presence or absence of TGF-beta blockade for 2 days, and then she selected the NK cells and tested their ability to kill GSC targets. And here you can see that whereas NK cells that were cultured with GSCs in the absence of TGF-beta blockade now became dysfunctional if a TGF-beta antibody was included that prevented the NK cells from becoming dysfunctional. The question then was, do these GSCs produce TGF-beta constitutively, or is this driven by a crosstalk with NK cells? So then what Hila did was she cultured NK cells either alone, GSCs, or with GSC cells in direct contact, or separated by a transwell. And what she demonstrated was that TGF-beta is only released when NK cells are in contact with GSCs. When they're separated with a transwell, the TGF-beta production is prevented, suggesting that there is probably a cell-cell contact, a dependent mechanism that is driving TGF-beta secretion by GSCs after contact with NK cells. And indeed, when the NK cells are separated from the GSCs by a transwell, they still maintain their ability to kill targets. So then they don't become dysfunctional. So we did a lot of various blocking experiments that I don't want to bore you with to try and find out what are those cell-cell contact molecules on the NK cells and the GSCs that drive really this crosstalk. And basically what we found was that the integrins on the surface of NK cells interact with actually the TGF-beta that is sitting on the surface of GSCs in an inactive form, and upon direct cell-cell contact, these integrins drive the release of TGF-beta from its inactive form to an active form and lead to suppression of NK cells. And this led to really an intriguing question to us, is whether we can protect our NK cells from this effect therapeutically, either by inhibiting this interaction of NK cell integrins with the TGF-beta lab, or by using CRISPR gene editing to knock out the TGF-beta receptor and therefore make NK cells no longer susceptible to the effect of TGF-beta. Or another alternative was to use a TGF-beta receptor kinase inhibitor called galunisertib that was in trials at the time that we did these in vivo experiments. So Mara Shanley in the group, who now leads the glioblastoma program, did these in vivo studies in collaboration with with Fred Lang and Joy Gumin, where NSG mice were implanted with GSCs, and then they received intracranial NK cell infusions, either with Silengitide, which is the inhibitor of the integrin interaction, or with galunisertib, which is the TGF-beta inhibitor. And here, as you can see, neither Silengitide or galunisertib alone could control the tumor. Actually, NK cells were doing a pretty good job of controlling the tumor if administered alone, but when administered with Silengitide and galunisertib, they really controlled the tumor extremely well. And this shows the Kaplan-Meier curves demonstrating that NK cells together with galunisertib or with Silengitide led to very nice survival of the animals. Peripheral blood NK cells, even without TGF-beta inhibition or integrin disruption, also could control the disease, not quite as well, but the other control groups, really, the animals succumbed to disease. Then Myra and Rafet decided then to test if TGF-beta receptor knockout using CRISPR could also protect NK cells from this effect. So here you can see very efficient knockout of the TGF-beta receptor 2. And the TGF-beta receptor knockout NK cells are now capable of maintaining their function compared to NK cells that didn't have TGF-beta receptor silence. And again, in vivo, Myra here demonstrated very nicely that when these animals then received intracranially TGF-beta receptor 2 knockout NK cells, they could control the tumor very nicely, better than animals that received NK cells that were wild type or NK cells in combination with galunisertib. So we're very excited, and this is the summary of the bioluminescence. So we're very excited about these data, and in collaboration with Amy Heimberger, Sharpe Weathers, John de Groot, and Fred Lang, and with the support of hopefully glioblastoma moonshot, what we would like to do is test these TGF-beta receptor 2 knockout NK cells for the treatment of patients with glioblastoma, so that we can take, again, umbilical cord blood, expand them, knock out the NK cells using gRNA, Cas9 RNP, and this will be done in our GMP facility, and then administer them to patients with relapse or refractory glioblastoma. And these studies will be supported, obviously, by extensive immune correlatives. So then, just to conclude the second part of my presentation, we've also shown that we can target checkpoints in NK cells, and that can increase current NK cell activity. I showed the example of CISH, but we are looking at other checkpoints as well, and engineering NK cells to target other cancers, and other antigens are in development. So with that, I have lots of people to thank. I mean, this kind of work really requires a village. So I'd like to thank E.J. Schwal, who's been a great collaborator, and she leads both our Cord Bank and MD Anderson GMP facility. Our department, Dr. Champlin, our head of department, our research nurses, our cord blood bank, our GMP facility, that are doing an amazing job of really expanding and developing these very complicated protocols of NK cell engineering. Our collaborators, and I should also mention, I've already mentioned also our glioblastoma collaborators in the departments of neurosurgery and neuro-oncology, and the very talented postdocs, scientists, technicians in the lab that do the work. Our funding agencies, and also the Moonshot programs. Thank you very much for your attention.
Thank you very much, Kati. Wonderful talk. It was really excellent. Let's start with the question. First question. Does using steroid affect CAR-NK cell therapy?
Yes, very good questions. NK cells, just like T cells, are susceptible to the lymphocytotoxic effect of corticosteroids. And that's one of the reasons that actually we've developed also glucocorticoid receptor knockout strategy that will be going to the clinic very soon with viral-specific T cells. So Rafet and May have developed this protocol, and ultimately with glioblastoma, maybe the best approach would be to do a multiplex gene editing to knock out both the TGF-beta receptor 2 and the glucocorticoid receptor. And this strategy, actually, Rafet Basar in the lab has already developed the protocol for that.
Thank you very much. Another question. How do you take a portion of the frozen cord blood unit for donor screening? And do you have screening criteria in addition to KIR mismatch?
Yes, so at the moment we are trying to go very agnostic with the cord blood, because ultimately we want to have an off-the-shelf product where you don't want to actually even worry about HLA matching, KIR ligand matching, etc. We are looking at the data with KIR ligand mismatch for the population. For only about 50% of patients can you find a donor that's KIR ligand mismatch. So ultimately we're gathering all the data, but we are not specifically going and picking a donor based on a set of criteria.
Thank you, Kati. Another question. What's the advantage of negative NK selection versus CD56 selection?
Yeah, that's a very good question. We started CD56 negative selection because we didn't know how CD56 positive selection would modulate the function of NK cells. Actually, we're now doing some of these head-to-head comparisons to see whether it matters. For GMP translation, there are no GMP grade CD56 beads. So we're kind of stuck with the negative selection because we have to use obviously GMP grade beads that are available for CD19, CD3, and CD14.
Thank you. Now, two questions who are correlated. So one participant asked, what would you consider as a solid cancer as best for CAR-NK therapy? And I would add except glioblastomas that you show wonderful data. And I combine with the second question. Why not to use CAR-NK in metastatic gastric cancer patient with ascites, since ascites cells contain lots of NK cells?
Okay, very good questions. So solid tumors, we're now very interested in solid tumors, and we've started collaborating with Fundameric to look at breast cancer, with Anaban Matra to look at pancreatic cancer, with Scott Kopetz to look at colon cancer. So, you know, certain cells certainly are more susceptible to NK-mediated killing. We have to study the tumors separately. What I can tell you, for instance, is with pancreatic cancer, we see that there is NK cell infiltration, and that gives you a clue that maybe NK cells will be good at targeting this type of cancer. The same with hepatic metastases. But for instance, with colon cancer, infiltration by NK cells is less. So now we have to try and understand, you know, what dictates homing, and we're looking at the different homing receptors and chemokine receptors that could then dictate the NK cells to get to colon cancer. Once we know what are the homing receptors that can help with that trafficking, then we can engineer our NK cells to express that particular homing receptor. With regards to the ascites, so I didn't go through the data, but the problem is the NK cells for patients with cancer are very dysfunctional, and they are very and they are they're not cytotoxic. So we can't use those autologous cells, but there is no reason to think that we cannot give allogeneic NK cells peritoneally to patients with gastric cancer or ovarian cancer. Having said that, we haven't done those experiments, but again, these are areas that we're very interested in exploring. I think now that we've shown proof of principle, NK cells are safe, NK cells can be engineered, and NK cells are effective, and with all the tools that we have to hand with engineering, with the ability to engineer the product, access to our cord blood bank, having the GMP facility that has the expertise to very rapidly translate these approaches to the clinic, we can move very rapidly and collaborate with the different groups in the institution to really develop these programs.
Thank you. And you got very nicely also the last question, which is why own NK cells, I suppose from patients, not working after NK cell therapy? If you want to add something to your last question, who answered, who was covering basically this.
Yeah, so tumor cells have developed very sophisticated mechanisms of escaping NK cell immune surveillance, and we've looked at that in multiple different cancers. And it seems to be a sort of global effect in that the NK cells that infiltrate the tumor often have upregulation of inhibitory receptors, downregulation of activating receptors. Many of the adaptive proteins and the transcription factors that dictate NK cell function are missing. So, and actually, even if you take those NK cells out, and you try and activate them and grow them in vitro, you can only marginally improve their function. So what is happening seems to be irreversible, and we want to try and understand why that's the case. We wonder whether it's just epigenetic modification of the cells that means that they are now becoming irreversibly altered, and they acquire this inhibitory phenotype.
Thank you very much, Kati. And with this, let's basically thanks to everybody who was present here. It was a wonderful talk. You guys are wonderful being with a large family of MD Anderson. And I want only to say one more sentence. Keep all in mind during this tough time that science has the power not only to discover, but science has the power to unify people. We love science because science is bringing us all together. Doesn't matter from where we come, doesn't matter of race, and basically science means to be together, to make discoveries, and to move the humanity forward. So thank you for all the big family of doing this. Thank you to Kati Rezvani and to all the people who give talks in Enjoy Science, and see all of you next Friday. Thank you again.
Thank you, everyone. Bye-bye.
Thank you, Kati.